Hi audience. So this is the next part of the series where I'm explaining the question number 1 to 5 of biotech section of GATE Biotechnology 2020 question paper, right? So the first question is protein P becomes functional upon phosphorylation of serine residue. Replacing this serine with some other amino acid will result in phosphomimic mutant of P. Okay, so this is the structure of serine. Yes, and so I'm making this. This is the alpha carbon. This is the hydrogen. This is the amine group. This is the C carboxyl group, and this is the R CH2 OH of serine. Okay, now see uh, what happens actually in phosphorylation. We add this, sorry for the structure, there is no space, that's why I'm the bonds can be seen distorted, but this is basically due to less space I have. Okay, so this is the additional group instead of H. See, O is here. So what do we see? We see the bulky group, addition of bulky group in the phosphorylated serine, and we see additional negative charge in the phosphorylated amino acid. Okay, so remember these two properties. Okay, now see the option. The first option, alanine. If that means if we substitute serine with alanine, it will be functional always. Is it the case? No. Alanine has CH3 group in R. Okay, so I've said now alpha carbon H amine group, carboxyl group, and this is the R group. So we don't see the bulkiness, we don't see the negative charge, so this can't be the right option. Sorry, I guess I hope you understood the question, and then I'm reading the option. Okay, so I'm again explaining the question. So it is said that the protein P have one serine residue, and when it is phosphorylated it becomes activated or functional. Okay, and we mutate if we mutate this one to some another amino acid, it will mimic phosphorylated serine, so it will be always functional. Okay, right. Now if we see the aspartic acid, see this is the structure of aspartic acid. We see here the COO minus. Yes, that means this have one negative charge and some bulky group, less bulky, but yes, it's less bulky, but bulky, so it is similar. Then phenylalanine, do you remember the phenylalanine? This is the R group, CH2. This is the R group in phenylalanine. So this has the bulky group but not the negative charge. Yes, you remember the bulkiness and negative charge is necessary to mimic the phosphorylated serine. So it has not the negative charge. Now see the next option. Lysine. Do you remember the lysine has lysine is a basic amino acid. Basic amino acid, so it has plus charge instead of negative charge. So this surely cannot be the answer. So what is the right answer? B. Aspartic acid, the right answer. Now moving to the next question. Next question is Rasp protein is a okay. So it says what is Rasp protein. So if you remember the MAP kinase pathway, you had studied the Rasp protein. What is Rasp protein there? So it is a GTPase. Say it is a cell membrane. Then in the inner side there is a protein which is cell surface attached called a Rasp protein. It is a GTPase, but it is a monomeric GTPase. Remember, you must have studied the different. There are GTPase which have alpha, beta, and gamma subunits. But this is an exception which has monomer. Yes, it is a monomer, and when signal binds to the surface receptor, it relays the signal from surface to cytosol or nucleus, okay, or the corresponding organelles, right? See what is the right answer. See, the first option, trimeric GTPase, no. Second option, monomeric GTPase, yes, it can be involved in relaying signal from cell surface to nucleus. Yes, this can be true. Third option, monomeric, no, sorry, trimeric GTPase, no. Fourth option, D, monomeric GTPase, yes, involved in regulation of cell cytoskeleton, no. It is involved in what do you say, cell proliferation. So sorry, you must have studied MAP kinase pathway in cell proliferation or apoptosis. So the actually the right option is B. Okay, now go to the next question. Next question is question number 3. Which of the following statements are correct? The first statement, virus can play a role in causing human cancer. Yes, yes, it's true. We will study in detail in the next slide actually. Tumor suppressor gene can be turned off without any change in its DNA sequence. Yes, it's true. Do you remember the epigenetic change? Epigenetic. Or do you remember the regulators like promoter, insulator, insulator, and enhancers, and there are many more things. So every term, insulator, promoter, enhancer, need to be explained, but in this video I will not explain every terminology, but I will give an idea that of the answer, that epigenetics or promoter, enhancer, etc can be used to turn off the tumor suppressor gene. Okay, now alteration in mi expression level contributes to the development of cancer. Yes, true. Actually, yeah, let me. So actually, I'm in IISER Kolkata. Okay, so IISER Kolkata. Hmm. Then there is a professor PSR. We call him PSR. Partho Sarthire. Partho Sarthi Ray. So he's working on a MIR 21, which has a role in cancer. Some specific cancer I don't remember. So I'm in different lab. So yes, it's true. So all the three options are true. So D is the right option. Okay. So in the next slide you can study the details. Okay. Now moving to the next question, that is, which which class of antibody is first made by developing B cells inside bone marrow? So it is a fact that IgM is the first antibody which appears on the surface of B cell. Also, IgM is the first antibody no, which you can see against any antigen in the primary response. Okay, so this is the D is the right answer. Now, fifth question. Determine the correctness or otherwise of the following assertion and reason regarding the mammalian cell. Okay, assertion is cells use calcium, not sodium, for cell to cell signaling. Yeah, true. Do you remember the neuromuscular junction or neuron to neuron attachment? It is neuromuscular junction and neuron to it is NM junction, and it is neuron to neuron. Okay, calcium is involved in passing signals. So here basically acetylcholine is used. Okay, that's the detail part, and yeah, we will see in another video or in regular classes. Now, the next question: in the cytosol, concentration of sodium plus is lower than that of calcium plus in the cytosol, inside the cell. Yes, inside the cell. Inside the cell, what is said? The concentration of sodium is lower than that of calcium. Actually, inside cell, all the calciums are stored in sarcoplasmic, sorry, um, ER, in endoplasmic reticulum. Okay, and that, but this is not necessarily true, because when it is stored somewhere in other organelle, then the concentration in the in the cytosol is lower. Okay, so it is not true. So here is the detail: concentration of calcium in the extracellular fluid, means outside the cell, is greater. Okay, outside the cell is greater, not inside. So this is wrong. So the option is A and R both true, no. A and R true, no. A and R false, no. A is true but R is false, yes. D option. Right? Now going to the sixth question. Vincristine, sorry, vincristine and vinblastine. Vin, vincristine and vinblastine, two commercially important secondary metabolites from Catharanthus roseus are example of. I remember this. These are alkaloids. These are alkaloids. Yeah. So this is the plant actually. This can be seen anywhere in outside home. So this is the flower. This is the flower, white, pink, purple. Uh, and here you will see the dark coloration, and below this you will see the stalk. Below this circle you will see the stalk. So this is very common flower, vinca rosea size. Yeah, vinca rosea. So these are alkaloids. Okay, and yes, it is used in treatment of many disease like Hodgkin disease, leukemia, and yes, both are neurotoxic. Yeah, you can read this. Now the seventh part: DNA synthesized from an RNA template is called. So remember the reverse transcription, RNA to DNA. This is the reverse transcription, but you will not see the option reverse containing reverse transcription. The first option is recombinant DNA. So do you remember what is recombinant DNA? Recombinant DNA is a DNA made up of two different DNA. So this is the one DNA, and this is the another. Yes. So this is the recombinant DNA. It is recombinant. Uh, you can see two inserts here, and this is the recombinant DNA. Now, transcript. Do you remember what is transcript? When DNA is transcribed to RNA, the RNA is called transcript. RNA is a transcript. Okay, so let me change the color. Hmm. tDNA. Okay, tDNA is a transfer DNA, transferable DNA. You will see in TI or RI plasmid. Okay, so I guess 11th question is about TI plasmid. Okay, sorry. 11th question is about TI plasmid. You will see there what is tDNA. Okay, now the next question is complementary DNA. Oh, sorry, complementary DNA. So cDNA or complementary DNA. So what is the use of cDNA? What is cDNA? I see, when DNA is transcribed, transcription is a process, to RNA, okay, then it contains both uh, exon and intron. Oh, yeah, intron. Intron are parts which are not expressed in the form of protein. Intron. Okay. So what happens after that? It needs further processing. So here is the RNA, which is smaller than the previous one. This one smaller. This is smaller than this one. Okay, and yes, this intron. So this is axon. This is intron. Okay. Now, uh, I rub this. This will be messy, actually. Yeah. So what was exon? Exon. So when exon is again reverse transcribed to DNA. So remember this, as this exon is smaller than the previous RNA, pre-processed RNA. So this DNA called cDNA, complementary DNA, sorry T, and it will be smaller than the original DNA because original DNA had introns. Okay, remember. So the sequence which contribute, which was processed, which was transcribed to the intron actually. Yes. So this is cDNA. So what is the advantage of cDNA? cDNA is stable, more stable than RNA. Yes, and cDNA can be used to store only usable, only required functional sequence, right? So that's why it can be used in cDNA library. So you will study this in biotechnology cDNA library. Oh, okay, okay. So you can understand now. Now the next question is, two monomeric his-tagged proteins of identical molecular weight are present in a solution. Okay, two proteins having the same molecular weight, and they have his amino acid. Okay, pi of these two proteins are 5.6 and 6 point, means they are different, they have different pi. Okay, which one of the following technique can be used to separate them? Okay, so we need to find a technique that can separate two proteins having the same molecular weight but different pi. So different pi different means different pH can be used to find the what do you say, ionic strength or the neutral amino acid, neutral protein. Okay, right, good. So for this, we need to emphasize on the iron part. We need denatured polyacrylamide. No, polyacrylamide gel for this is no denatured page. This is very long. Page is the page no. This separate the uh, protein only on the basis of molecular weight. Protein or anything only in the molecular weight, not charge. Size exclusion chromatography, no, no size, no. Ion exchange, yes, it can be used. Nickel affinity, yes, it can be used. Why? Nickel has the capability to form coordinate bond, so it can be used. But more appropriate or the more better, like more mm, accurate answer is ion exchange chromatography. How? So I'm showing. So in ion exchange chromatography, there is a technique we use when matrix has immobilized, say, nickel, not only nickel, copper, or any what do you say, the d block element, yes, that can form the coordinate bond. That's why I said the nickel affinity chromatography can be used, uh, right? And then when the mixture is passed, the mixture will form the coordinate bond. So, it will be given a fixed pH. Huh. The one will form coordinate bond and another will pass, and you will get the two thing. Both will pass, but at different time. Okay, so this is the right answer. Yes, you can read the details actually. Yes, why he the actually histidine amino acid makes coordinate bond. Why? Because of the presence of imidazole ring, which has nitrogen plus, sorry, nitrogen and lone pair of nitrogen. So this lone pair can be transferred. So you can read somewhere. Yes, histidine's imidazole ring. Hmm? Okay. Now the next question. Next question is, during a positive negative selection process, transformed animal cell expressing blank are killed in presence of canciclovir. Okay. Hmm. What is ganciclovir? Do you know? So ganciclovir um, inhibits the replication, especially the herpes virus. Okay, inhibits the replication of herpes virus. How would, so that's why if transformed DNA has say something, some insertion which what do you say, which inactivates, sorry, I have to read the question again. Presence of glancewear transformed cell. some insert which so that it does not form. Transformed tin has some insert so that it does not form the necessary um, equipment to stop the mechanism of Glancevier. Okay, yes. uh, yes. So in the transformed cell, sorry, this is the transformed, right? And it, due to the presence of insert, it does not form something I don't know. Hmm. So nothing is said about that. Does not form a thing which will stop Glancevier, which Glancevier, it does not form. This doesn't happen. So actually, what let me say the detail of how what is ganciclovir works. So ganciclovir is inactivated form. It is activated by the phosphorylation, by the addition of triphosphate. Okay, so ganciclovir first gets monophosphorylated viral by the viral thymidine kinase. Okay. Hmm. Uh, when viral thymidine kinase is there, suppose DNA, sorry, DNA can form thymidine kinase TK, then ganciclovir tablet I made, ganciclovir, it will help activate, it will activate ganciclovir, okay, by phosphorylating. Then another enzyme will be needed to try phosphorylate the ganciclovir, okay. And then what will ganciclovir do? It will inhibit the replication of DNA. Okay, got it? I guess I'm now clear now, in the later part, not the earlier part. Okay, so right answer is pyruvate kinase no, viral thymidine kinase yes, and rest you don't need to see. Option D. A vector derived from which one of the following virus is used for high frequency genomic interaction integration of a transgene in animal cells? Option A, adenovirus. Yes, it is used, but efficiency is less, or if the overall the efficiency, stability, and the immunogenicity is not favorable. You can say adeno-associated virus, it is much better, but yeah, lentivirus is best among all this. Yeah. So next part, I had said actually about the TI plasmid in the previous question. Do you remember? Yes, tDNA, tDNA. I had said no, 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 no. 11th question. Which one of the following statement about agrobacterium TI plasmid is correct? Okay. Option A. Weir genes are located within the tDNA segment. So you can see this is the tDNA region, transferable region, which can be transferred from the virus to plant cell. Okay. Virgin, no, no. In the tDNA segment, there is no virgin. So not true. Phytohormone biosynthesis genes are located outside the, no, you can see. cytokine, auxin. So these are inside the T tDNA region. The next question: opine catabolism genes are located within the tDNA segment. C. Opine catabolism. This is outside the tDNA segment. So no. So all three wrong. Fourth option: opine biosynthesis gene are located within the. Yes, you can see the right option. The fourth option. Now go to the next question. That is 12th. Which one of the following types of molecule act as a biological catalyst? Yes. So which can act as an enzyme? One, protein. Yes, enzymes are generally proteinous in nature. RNA. Yes, RNA is. Yes, RNA can you can actually see where self splicing. Yes, self splicing. And actually, when you see self splicing, you should know splicing is of two types: group 1 and group 2. I'm talking about group 1 and group 2. Okay, you can study. And the phospholipid, yes, phospholipid, no, phospholipid cannot catalyze the reaction, no. So you can read the detail. The right option is P and Q only A. Now the next question: which one of the following media component is used to maintain the pH in mammalian cell culture? Okay, first, calcium chloride. No, it is used as a source of calcium, and again magnesium as a source. Sodium chloride is a source. Yes, sodium bicarbonate. It is a buffer also. It also contains NaHCO3 and Na2CO3. It has both, but in different ratio to maintain the different pH. Hmm. So the right option is D. Now going to the next question. Next question is which of the following are energy transducing membrane? Plasma membrane of bacteria. Yes, if I remember, there is a mesosome structure, hmm, which is involved in respiration. Okay, and then inner membrane of chloroplast. Actually, inner membrane of chloroplast. Uh, sorry, no. Chloroplast is double membrane, and you will see F O and F1 particle in the thylakoid membrane. So thylakoid membrane can be third membrane. No, this is wrong. Inner membrane of mitochondria. Yes, you will see there inner membrane of mitochondria. Yes. So P and R only. The option B is right. Now going to the 15th question: amino acid sequence of cytochrome C and ribulose 5 phosphate epimers from 40 organisms were chosen. Okay, and phylogenetic trees were obtained from each of these two protein families. Okay, determine the correctness or otherwise of the following assertion and the reason. Okay, the two no, sorry, assertion is the two trees will not be identical. Okay, it's true, they are not identical. Why? Because if they are living the same condition, they have different capability to sustain everything, like small, even minor mutation, or small even types of mutation, or mutations are random. See, mutations are random, so anything can happen, anything can happen. So no. The reason is nature and the frequency of mutation. Yes, nature of the mutation, which type of mutation, like insertion, addition, or addition, like what do you say, deletion, something, or the frequency. Yes, how many times it mutated, both can be the reason. Yes, both are true, and the exact reason, region is the exact region. So option A is the both A and are true, and that is the correct region. Right answer. Going to the 16th question. A microorganism isolated from a salt rich, sorry, the salt rich, the salt concentration is given nearly 2 molar lake. Okay, found to possess diglycerol tetraethers with Baba. So that's a word. Polyisoprenoid alcohol side chains as a major lipid component of its cell membrane. Isolated organism is one polyplanktomycete. So first, when it is isolated from rich uh, lake which is rich in salt, that means the bacteria is a halophile. Okay, so halophile means if you remember class 11th, then it is what do you say, the archaebacteria. Yes. Mm, and then this one diglycerol. So this is the characteristic of archaea, all archaea. It's the characteristic of archaea. So the option planktomycete nahi, cyanobacteria no, unicellular amoeba no, archaea is right. Yeah. Going to the next question. The question is function f is as follows. Okay, function fx equals to 15 if x is lower than 1, less than 1, and fx equal to cx if x is greater than or equals to 1. Now, if function f is continuous, then what is the value of c? No, sorry, I'm saying c. Yes, continuous function. Sorry, I need continuous function. If one ends here, say 15 is approached, now something will start from the same point, continuous. Otherwise, in discontinuous function, if it ends at 15, then something will start, let me, yes, something will start from somewhere else. So there is a discontinuity or gap. Okay, so as it is given it is continuous, so we need to equate equate uh, to 15, equate to 15, so that the minimum 15 can be get to make it continue. Okay, so it is cx equals to 15. Suppose x is 1, suppose x is 1, then the value of c equals to 15. Okay, if x is greater than 1, whole number, whole number, x is 2, if x is 2, then c equals to 7.5. No, it cannot be. If c equals to 3, then sorry, x equals to 3, then c equals to 5, right? uh, if x equals to x, 1 said, 3 said, sorry, and 5, 5, c equals to 3, 15 by 5. If x equal to 15, c equals to 1. So anything can be the answer. 1, 3, 5, 15. Okay. Now going to the next question. Next question is given that z equals to x square plus y square, the value of Deber z by Deber x for x equals to 1 and y equals to 0. Okay, so I need to find delta z by delta x when x equals to 1 and y equals to 0. Okay, so I need to first find out the differentiation. Mm, so it is given dz by dx equals to dx square by dx plus dy square by dx. See? Oh, so here is a minor mistake. Here is a minor mistake. See, I said dx. I have written x only, sorry. Now, this is equals to 2x. So, this is the formula. Say dx square by dx equals to x 2 minus 1 into 2 minus 1 equals to 1. Sorry. No, sorry. Equals to 2. Sorry. 2. The the value 2 into x. This is multiplication sign. X is, let me into d x square by dx equals to, as per formula, 2 into x, the given power 2 minus 1 equals to 2x, right? So the value of dx square by x is 2x and dy square by dx. So y is not changing. Y is not dependent on x, right? Y is not changing. Y is not changing because of the change in x. And also y equals to 0, so it is 0. Second part. Now if x equals to 1, so dz x by dx equals to 2x, and x equals to 1, so it is 2. Answer. Okay. Now the next question. The elemental composition of dry biomass of yeast species is itna. So long. The contribution of carbon to the dry biomass is so easy actually. So what do you need to do? You need to uh, actually, if it is percentage, so yeah, actually, no, instead of this, uh, this should not be here. This should be here. Hmm. Something was written. I need to check again. I forget. Yeah, 1200 by 23.4 equals to 51.27. Now this is the solution. So what do you need to do? Uh, okay. Uh, Actually, this should not be here. There are mistakes. Atomic weight of biomass. See. Now, suppose, how do you solve this? Suppose this is the complete series, okay, and you have only this part. Then I ask, what is the contribution of this part? What will you do? You will yellow divided by red. You will do this. But if I ask, what is the contribution of mass of yellow? Then you will write mass of yellow divided by mass of red. Okay. This is the only thing I have done. But when it is in percentage, then it is multiplied by 100, right? So this is the actual concept. Now you can see we have done nothing, atomic weight of carbon, that is 12, divided by atomic biomass of the atomic weight of biomass into 100. That is simple. Simple. There is nothing chemistry, nothing science. It is only common sense, or child. You can learn this in childhood. Actually. Uh, Sorry, you're right. Solvent A and B are completely immiscible. Okay. Solute S is soluble in both these solvent. Okay. 100 gram of S was added to a container which has 2 kg of each A and B. Great. It is 2 kg. Remember, the solvent is given in kg. The solute is 1.5 times more soluble in solvent A than in solvent B. The mixture was agitated thoroughly and allowed to reach equilibrium. Assume that solute has completely dissolved. The amount of solute in solvent A phase is okay. So actually it is solved. C. Uh, Actually, suppose x gram of solute hmm, is um, in phase B, in solvent B, hmm, and solvent then 1.5x gram will be in solvent A. This is in B and this is in A. Right? Now, what is the sum x gram plus 1.5 x gram will give total weight, that is the 100 gram of the solute. Now just solve this. 2.5x equals to 100, and when you solve this, x equals to 40 gram. Answer. So this is the 40 gram is dissolved in solvent B. Solvent B. And what is the question? Question is solvent A. What is the dissolved amount in solvent A? Then 1.5 into 40 equals to 60. Answer. So till now, no, let's move. See the 21. Uh, the number of molecules of a nucleotide of molecular weight 300 gram per mole present in 10 picomole is uh, okay. So if molecular weight, so it is given, sorry, everything is given. Molecular weight equals to 300 gram. 300 gram contains how many um, Na particles, right? 6.02 into 10 to the power 23 particle, right? Hmm? And what is the number of nucleotides present in 10 picomole? So picomole equals to 10 to the power minus 12, right? 10 to the power minus 12 mole. So I should have written mole here. Hmm. Then how many particles should be present in 10, 1 picomole, right? So see, 300 molecular weight, 1 mole Na particle. Hmm. 10 power minus 12 mole equals to Na into 10 to the power minus 12 particle, right? that is 6.023 into 10 to the power 23 into 10 to the power minus 12. What is the answer? 6.023 into 10 to the power 11. 11, see, 23 minus 12, 11. So this is the answer. This is the answer. But answer is asked in 10 to the power 12. So erase this. The right answer is 6.023 into 10 to the power 11. Right? In 6.023 into 10 to the power 11 into 10 by 10, so I get zero point 0.6 into this 10 to the power 12, right? Right answer. Hmm. Sorry, 10 picomole, not 1 picomole. It is about 10 picomole. Sorry. I made mistake. 300 gram, 1 mole, and a particle 10 into 10 power minus 12 mole Na into 10 power minus 11. Hmm. Minus 11, right? That is 6.023 into 10 to the power 23 into 10 power minus 11. So answer will be, may erase all, answer will be 6.023 into 10 to the power 12. Correct? So the next part, in the next part we will discuss the next few questions. Okay, till then, thank you.